All jurors answer in the negative.
Have you discussed this case with anyone?
All jurors respond in the negative.
Have you permitted anyone to discuss this case with you?
All jurors respond in the negative.
Have you conducted any research into this case?
All jurors respond in the negative.
And finally, has anybody persisted in attempting to discuss this case with you?
All jurors respond in the negative.
All right. Well again, ladies and gentlemen, thank you for continuing to comply with the court's directives. We are ready to continue. Mr. Luttrell, you may call your next witness.
Yes, Your Honor. Thank you. The state would call Stacy Bozinovski.
Witness sworn.
Would you state your name, please, and spell your last name or first and last name?
My name is Stacy Bozinovski. My first name is spelled Stacy. My last name is spelled Bozinovski.
Where are you working?
I am employed with the Indiana State Police Laboratory in Indianapolis.
What is your profession?
I am a forensic scientist, or more specifically, a DNA analyst within the biology section at the laboratory.
Would you tell the jury the role of a forensic scientist?
My role as a forensic scientist is to perform a serological examination of evidentiary items for the presence and identification of body fluid stains or other sources of biological material. I then attempt to develop a DNA profile from those sources through DNA analysis. I compare profiles from unknown evidentiary samples to profiles from known standards of individuals that may be involved in a case. The comparison will allow me to include or exclude individuals as possible contributors to a sample. Also, as part of my role as a forensic scientist, I primarily perform casework using autosomal STR technologies. However, I perform casework using other technologies, including YSTR analysis and next-generation sequencing, which uses SNP analysis. I'm currently involved with an internal validation at our laboratory to bring on an additional technology that will bring forensic investigative genetic genealogy to the Indiana State Police Laboratory, and then that will allow us to use that technology on some of the most complex cases in our state. In addition, I specialize in paternity analysis or kinship analysis, which is used in unidentified human.
So you're in a field that is ever-changing?
That's correct. There's a continuous updates and technology in the DNA field.
And your career is an expression of that, of how you have added specialties your area of serology and DNA. Is that correct?
That would be correct.
What's your educational background?
I have a Bachelor of Science degree in biology from Indiana University in Bloomington, and I also have a Master of Science degree in forensic science from Marshall University.
And I think you've touched on some of this already. But have you obtained specialized training in the area of forensic biology? And could you explain what that additional specialized training has been?
Yes. As part of my training with the Indiana State Police Laboratory, I've completed training in both serological examinations and DNA analysis. This training included several sections about the identification of body fluid stains and also the theories and methods of the different stages of DNA analysis. Each training consists of various combinations of lectures, scientific readings, demonstrations, and training exercises, and I also have successfully completed examination of unknown samples, oral reviews, and written examinations as part of my training.
And do you belong to any professional organizations that you can draw upon and help you do this job?
Yes, sir.
Would you tell the jury what those are?
I'm a member of the Midwestern Association of Forensic Scientists, as well as a member of the American Academy of Forensic Sciences. I've received certification in the field of molecular biology with the American Board of Criminalistics.
Why are those important?
It's important to be members of scientific organizations in our field to expose us to new technologies, new information that's ever present in the field of DNA analysis. My certification in the field of molecular biology also is just a voluntary process that means I have been qualified by a group of my peers as competent and proficient in that field. It's completely voluntary, not mandated by any agency to perform the science. it's just an extra credential.
And is the Indiana State Police Laboratory accredited?
Yes, sir.
And by whom?
We are accredited by an organization called ANAB, which stands for ANSI National Accreditation Board. ANSI is an acronym that stands for American National Standards Institute.
And why is that important?
The accreditation process ensures and shows the reliability and accuracy of our scientific work. So in order to complete accreditation, we have a team of qualified individuals come into our laboratory and monitor all aspects of the laboratory, including management, operations, procedures, equipment, and then they will assess all OFR protocols, make sure that we're following the national standards in the forensic science community to receive accreditation.
I'm gonna hand you what's been marked as States Exhibit 283. Do you recognize that?
Yes, sir.
And what is it?
States Exhibit 283 is a copy of my CV or resume that I've provided.
And what's important about the information on that document?
Well, this document outlines my experience within my career. It also shows the information that we've just talked about as far as my education and professional organizations, but the majority of the outline of the document outlines the scientific training that I've undergone under the course of my 18-year career with the State Police Laboratory.
I would move the admission, Your Honor, of this document, the CV of Ms. Bozinovski, States Exhibit 283. States Exhibit 283 offered.
No objection. All right. Without objection, 283 is admitted.
Thank you, Your Honor. May I publish to the jury?
You may.
May we approach, Your Honor?
Yes.
Sidebar conference conducted.
We're gonna be talking about so many items.
Okay.
But we've broken them down by report, just so you know.
Okay. That's good for you.
I have the first report, which is what the staff is organizing, but I don't have all the second items ready.
Okay.
On that table. Can we have the staff help?
Sure.
Get those while she's testifying?
Sure.
Okay.
Of course.
I just wanted to.
Sidebar conference concluded.
Does the Indiana State Police Laboratory have a quality assurance/quality control process or program in place?
Yes, sir.
And why is that important?
The quality assurance/quality control program at our laboratory again helps ensure the accuracy and reliability of our results. It is a process that is used to help monitor procedures, verify that we're following all of our protocols in place, and to identify any problems that might be, and to take any corrective actions if problems are identified.
And is that all required by the accreditation? Is that one of the things that is demanded and expected of the agency?
It is one of the standards as part of accreditation to have that kind of protocol in place in your laboratory.
Are there any elements in that program that you haven't covered so far that you'd like to share with the jury?
Sure. I can give some examples of some components of the program to help put into perspective how that plays a role in our analysis. Examples of some quality control components would be the training of our analysts, the education that's required, as well, the internal and external audits that our laboratory undergo the validation of our analytical methods, so the procedures and the processes that we are using for analysis, then, the calibration of our equipment and reagents, those kind of things that happen in the laboratory. Some examples of quality control would be some internal actions or activities that we use every day as part of our analysis, so some examples of that would be the use of positive and negative controls to ensure that our reagents, our chemicals, are working appropriately, the wearing of PROP or personal protective equipment to protect ourselves and the evidence, the documentation that's required as part of our case analysis, the technical and administrative review process that each case undergoes, as well.
What's DNA and where do we find it?
So DNA stands for deoxyribonucleic acid. It's a biological material that contains an information code, so it serves as a blueprint for how our body is built, how it functions. DNA is passed on from generation to generation, so it's inherited. You inherit half of your DNA from your mother and half from your.
You mentioned your DNA work and you mentioned serology. Would you tell us the difference?
So as part of my role as a forensic scientist, I use both serological examinations and DNA analysis. So serological, or I guess serology, is defined as the study of body fluids. So a serological examination is an exam that I do to help detect/locate body fluid stains and then identify those body fluid stains, or I help look for other biological material. DNA analysis is a method that is used to help individualize those body fluid stains to help make conclusions on who may be consistent with that sample to help provide some meaning behind that body fluid sample.
What are examples of different sources of DNA?
So most commonly in a serological examination, we will see body fluid stains such as blood, seminal material, and saliva. Those are typically good sources of DNA. Other body fluids or materials like vomit, fecal material, and urine might be present on an item, but that is less common in the types of items that we examine, and those also are less optimal sources for DNA. In a serological examination, I may be looking for biological material like hair. Hair may be collected from an item to perform DNA analysis, as well, and this is also a less ideal source of DNA. And then finally, it is possible to collect or attempt to collect skin cells from evidentiary items, and the success rate of skin cells is highly variable on evidence, and it's often dependent on the quantity and the quality of that type of sample.
So at the top of this is a good to bad flow chart of sources of DNA. At the very top, good, or maybe okay to say rich source of DNA, would that be appropriate?
Yeah, I usually would say good, but a good source of DNA or rich in cellular material would be a blood-type sample.
Blood. And by good, you're talking about the quantity of DNA that's available in even a small amount of blood?
Yes, which kind of?
What do you mean by good?
Yes, which correlates to, essentially, the number of cells that may be present in that type of body fluid.
And what else is up there besides blood at the top of this flow chart?
I would assign seminal material and saliva and blood as all good sources of DNA.
At the bottom of that chart is skin cells?
Again, it's just highly variable. It all goes back to quantity and quality. So skin cells, the type of examinations that we do, there could be items that yield an abundance of skin cells, and there may be items that is hard to detect, anything, any DNA. And then, if I may elaborate, I don't actually have a test for detecting skin cells. It's just I attempt to collect skin cells of off items, and my tests to see if that was successful or not is based off the quantity of DNA present.
So when you are searching for and analyzing and finding DNA, in that process you're not identifying it as one of these types of DNA on the flowchart, it's just DNA. Is that correct?
Serological examination will help with yielding an identification on whether there's a body fluid or not. The DNA analysis results will just indicate what the DNA profile is. It doesn't indicate that it is blood. If that helps make sense.
And can you tell in your testing when DNA was deposited on the item that you examined?
No, there is not a test that I can perform that will, essentially, timestamp a DNA profile. That's not possible.
You used the word profile. Tell the jury what you mean by DNA profile.
A DNA profile is a list of numbers that makes up the genes or the markers that I'm analyzing in my analysis.
And when you are looking for body fluids, how do you do that? What processes did you do to identify body fluids?
So I will perform a serological examination in order to determine whether a body fluid is present. I first will do a presumptive test, which is a quick, simple, sensitive test it's a chemical test, so it requires the addition of 2 chemical components, and it may indicate that a body fluid is present. In order to confirm the presence of a body fluid, I will do a second test, called a confirmatory test, and that test is much more specific to the type of body fluid that I am analyzing and will confirm that a body fluid is present.
Is it too simplifying it to say the first stage of that test, you find out whether you might have a body fluid? The second stage is confirming whether you have a body fluid?
Yes. I also like to put it as the first test will screen out the samples that are not consistent with a body fluid.
Could you briefly describe the process that the laboratory goes through to get a DNA analysis?
DNA analysis occurs in several stages. The first step is called extraction, and that's where I add some chemicals. I take a cutting of a sample, add some chemicals to break open the cellular material and release the DNA. Then I will perform a step called quantification, and in that step I'll be able to determine how much human DNA and how much male DNA, as well as if any inhibition or degradation is present in a sample. If there's enough DNA present in the sample from that step, I'll continue on to the third stage, which is called amplification, and in that step I perform what's called a PCR, or a PCR, which is where I make millions of copies of specific markers or pieces of DNA that are of interest for our analysis. Those pieces of DNA are then separated by size using an electrical current, and this is called capillary electrophoresis. That separation is what allows us to produce the DNA profile. It yields a pattern that, essentially, I translate into that list of numbers or DNA profile.
There are some terms that we're gonna come across occasionally that I may stop and ask you to define or explain to us. What is a gene?
So a gene is a small, non-coding segment of DNA that we're interested in and in analyzing.
And what is an allele?
An allele is a variant or one of many different forms that may be expressed at that gene.
And what is a locus?
And a locus is the specific location of a gene on the DNA chromosome.
And why is DNA analysis performed?
Again, DNA analysis is, well, DNA in general is highly variable among individuals. We all have unique DNA profiles, and DNA analysis will allow us to individualize a sample or a body fluid stain to help determine who it may be consistent with.
I believe we'll be talking about today's single source and mixture profiles, correct?
Yes, sir.
And would you explain those to the jury, please?
I will use probably throughout my testimony the term single source and mixtures. Single source is terminology used to express that a DNA profile is consistent with a single individual, and so at each of those genes or markers, I am observing up to 2 alleles in the DNA profile, so that means I am able to confidently determine that it's coming from only one individual. It is single source. A mixture is a DNA profile that is coming from 2 or more individuals, and those kinds of DNA profiles you will observe multiple alleles at each of the markers that we're analyzing.
Are there occasions where you cannot determine the number of contributors to a mixture?
Yes, sir.
And what does that then pose to you as a DNA analyst?
Well, I think first thing we need to step back and talk about just how I go about analyzing a DNA profile, and essentially the first step is to assess for any biological artifacts that may exist in that DNA profile, and then the following step is to determine how many contributors may be present in a DNA mixture. So those determinations are made, that interpretation of the DNA sample is made.
We covered some of this, but what type of results may a DNA analysis generate?
So one of the stages that I mentioned earlier called quantification is a step where I can determine how much DNA, how much human DNA, how much male DNA is present, as well as other quality factors about the sample, like inhibition and degradation, that can all be assessed and determined during that stage. If there's not enough human DNA or even enough male DNA present in a sample, then that sample will not continue through the rest of the stages of DNA analysis, so that is a possible result that may be yielded from my analysis. Also, if there is enough DNA and it completes the DNA analysis process, then it may be suitable for interpretation, so I may be able to make determinations on the number of contributors and be able to compare it back to known standards in the case. And in that case, I will yield results indicating whether a sample, or whether a person could be included or excluded from a sample. Sometimes DNA samples cannot yield conclusive results. Again, based off of the quantity and quality, I may not be able to determine or make conclusions about a sample to make those comparisons back to known standards.
And were you asked, in this particular case, to conduct DNA analysis on a number of items that were presented to you, beginning in February of 2017?
Yes, sir.
And under what case number?
That case number would be laboratory case number 17K00066.
You have that number memorized?
I do.
Okay. And that resulted in you issuing a report on March the 7th of 2017, correct?
That is correct.
So we're gonna group and just look at that report items, then we'll move on to another report, other reports. I'm gonna begin by handing to you States Exhibit 174. I also have here 175. Are you familiar with both of these items?
Yes, sir.
And how are you familiar with them?
I recognize both of these items as items that I have examined in this case. I can recognize my markings for identification on the outside of the box, which includes my case number, my item number, my initials, and my personal employee number. Each item contains that information here, as well as my initials across the seal right here.
And you're familiar with the container and the type of items that are often included inside those boxes. Is that correct?
Yes, sir.
An Indiana sexual assault evidence collection kit is something you examined the contents of on May.
That's correct.
These specifically, Exhibit 174 is for Abby and is from Abby, and 175 is from Libby. And did you, in your process, go through each sub-item in these boxes and make an examination on them?
Yes, I did.
Would you tell the jury the results of your examination of those items?
Yes, sir. Your Honor, may I refer to my reports?
You may.
Thank you. So the contents of the evidence collection kits collected from Libby and Abby contained a series of items. Those items from each kit were vaginal wash samples, vaginal/cervical swabs, oral swabs, anal swabs, external genital swabs, internal genital swabs, fingernail scrapings. In addition, a head hair combing sample was collected from both Libby and Abby, and a pubic hair combing was collected from Libby. As I mentioned earlier, my analysis begins with a serological examination, so those items were tested and screened for the presence of seminal material. No seminal material was detected on the contents of the kits that were tested, with the exception of the vaginal/cervical swabs from each kit indicated the possible presence of seminal material, but there was no confirmatory testing performed. Moving on to my DNA analysis results, the contents of both of those evidence collection kits. In order, I did the extraction and the quantification, and, as part of the quantification step, I analyzed the amount of male DNA present in the samples, and the contents of the kits mostly exhibited no male DNA, with the exception of one set of external genital swabs from Abby and one set of external genital swabs from Libby, as well as one sample from the fingernail scrapings of each of the kits. Those samples indicated the presence of male DNA, however, there was an insufficient amount of male DNA for further analysis. So like I mentioned earlier, given that there was no male.
So back to the presumptive or preliminary result on both victims' kits where you had a presumptive positive, did I hear that correctly? For seminal material.
Yes, sir.
You said you did not do a confirmatory test, and why is that?
So at the time, we were not performing confirmatory tests as part of our workflow for sexual assault kits. Instead, it was a choice to help conserve sample, to take that sample forward for DNA analysis to determine what profile was present on the sample regardless of whether there was a confirmation of seminal material or not.
That presumptive, and then, if you had gone forward with confirmatory, that would have been a serological analysis, correct?
That's correct.
You wanted to concentrate, save the material for DNA analysis?
That's correct. So an additional serological test would have taken another cutting of that sample, and, in order to make the most use of time and resources, it was better just to go forward and see what type of DNA profile was present on that sample.
Have you ever had the experience where, on the initial test for seminal material, is followed by a confirmatory test and it's negative? Has that ever happened?
Are you asking if the initial test is positive and then the confirmatory test?
In this case, what was the result of your DNA analysis on those? You said they were both the cervical vaginal/cervical swabs on both victims.
There was, yes, the vaginal/cervical swabs of both of the kits from both individuals.
And what was the result of your DNA analysis on those swabs?
So the DNA result indicated that there was no male DNA present on both of those samples.
Now, the areas where you did get a quantity of male DNA, but not enough to develop a profile, did I say that correctly?
Yes.
One was on a fingernail of, was it Abby or Libby?
One set of fingernail scrapings from both individuals.
And the other was on an exterior external swab, external genital swab?
That's correct.
Does the finding of some male DNA on those swabs indicate that that would have been from a perpetrator of a crime?
No, it does not.
Would you explain that to the jury?
Well, I can't make any conclusions about who that DNA is attributable to, first of all, because I don't have a DNA profile, but DNA on fingernail scrapings and even external genital swabs, male DNA of the amounts that I was detecting, which was very, very little, isn't necessarily an unusual result off of those types of samples. The hands and fingernails are every day in contact with items and people, that it's easy to pick up a small amount of male DNA or female DNA from.
So it's if a victim were living within the same household with males, would you be surprised to find some quantity, insufficient quantity, but some quantity of male DNA in these locations where it was found on these two sexual assault collection kits?
I wouldn't be surprised. And when I'm speaking of male DNA, I want to remind you it was below the amount of male DNA we need to amplify to develop a profile, so it was a very, very little amount of male DNA, so it wouldn't surprise me that it could be coming from the environment or clothing or, like I said before, previously contacting objections or persons.
I'm gonna hand to you items 170 to State's Exhibits 170 to 173 and ask if those are familiar to you.
Yes, I do recognize State's Exhibits 170 through 173.
And in addition to those, I'm going to hand you to examine 151, 152, Exhibits 153, 154, and 155. Take a look at those and tell us if you recognize that packaging.
Yes, I do.
And how do you recognize them?
I recognize the packages again from my markings, which are included in a boxed area on each of the packages, including the case number and item number, my initials, and personal employee number. Each package has several of my initials across the seals.
If I might hand you these items, as well, State's Exhibit 149, 150, and 156, do you recognize?
Mr. Luttrell, what numbers were those?
State's Exhibits 149, 150, and 156.
Thank you. Sorry.
No.
Yes, I recognize these, as well.
All of the items submitted to you that you reported on in your May or March 7th report, have you recorded who from whom you received them?
Yes, that is recorded in a chain of custody maintained in our laboratory.
And can you tell the jury from whom you received each of these items?
Oh, thank you. So all of these items were submitted to the laboratory by Sergeant Brian Olehe, and I received the majority of them directly from Sergeant Olehe on February 17, 2017, with the exception of laboratory items 17, 18, and 27, which went for a trace examination once they were submitted into the laboratory, so I received them directly from that trace examiner after their analysis.
One other thing I want to circle back to very quickly, on these evidence collection kits, States Exhibit 174 and 175, in addition to doing an examination on unknowns, did you use any material in 174 and 175 to create a known standard?
Yes, and I appreciate you bringing that up, because I did hesitate in mentioning that I also did receive DNA blood standards from Abby and Libby as part of their evidence collection kit.
And how did you then use those blood collections that you received?
Yes. Those two blood samples were used as the DNA standard, so the known sample for Libby and Abby, throughout my analysis in this case.
Okay. Now I put you through a bundle of numbers and you have numbers, Sergeant Olehe had numbers, and now the court has numbers. Can I ask you to describe your results from the items that I've handed you and you've identified? How can I help you do that?
Yes, I would like to refer to those by laboratory item number, since that's how I have that documented throughout my case number, if that works.
Let's take these, if you're okay with this n number at a time, and then tell us, if you can, what your results were.
So first I would like to start with the laboratory items 1, 2, 8, and 9, which were States Exhibits 170 through 173. These were swabs collected from the body of Libby. Each of these samples were examined. A serological exam was performed for the presence of blood, and blood was detected on laboratory items 1, 8, and 9. Item 2 indicated the possible presence of blood. However, I did not do the confirmatory testing due to the amount of stain that was available. I wanted to preserve that stain for DNA analysis. In my DNA analysis of each of these 4 samples, I was looking for the presence of male DNA, much like I did with the components of the evidence collection kit. So they were extracted and quantification was performed and it was analyzed for the presence of male DNA. There was no male DNA detected on item 2, which was the swab of the right upper thigh. Items 1, the swab of the left thigh, item 8, the swab of the right breast, and item 9, the swab of the left breast indicated an insufficient.
The swab from the tree?
Yes, sir.
Does that complete? Did you go through each one of those?
I believe I got them.
If the record showed that the first group P of envelopes that you looked at, that followed your discussion of the collection kits, all came from the autopsy, that would be your item 100, 101, 107, and 108. Is that consistent with your documentation?
Those item numbers are referring to the agency item number, so that's the CSI's item number. My item numbers are items 1, 2, 8, and 9.
I'm sorry, yes.
No problem.
I've now confused the numbering system. States Exhibit 170, 171, 172, and 173 are what you had previously described. Did those all come? Were they all collected at the autopsy?
I believe that is the information that I have received about those swabs, yes.
Did you do further examination of the hair found on Abby's finger that was identified as an unknown female?
Yes. Given that it was a known profile, would compare that or use it for comparison if needed, yes.
And in examining that DNA profile on the strand of material, the hair, on the right hand of Abby, States Exhibit 149, were you able to make any conclusions about that DNA profile on that hair?
Well, right away, as I mentioned, I compared that DNA profile back to Abby and Libby as being the standards I had available for me at the time, and in doing that, I could tell you it was not consistent with either of their profile, but I did recognize that there was some similarities between that female profile and the profile of Libby, so that led me to believe that it could have been a biological relative of Libby at that time.
Did you later, Ms. Bozinovski, in March of 2017, receive what I've marked as States Exhibit 284, which is your lab number item 246? Did you later receive this?
I did receive this. I can't recall the exact date and I can refer to my chain of custody if you would like me to recall that, but I did receive this item. I have examined this item, and I recognize it again by my packaging, my markings on.
And what is that item?
This item is a DNA standard collected from Derek German.
Identified to you as the father of Libby?
That was the information that was provided me at the time that it was submitted to the laboratory.
I would move the admission of States Exhibit 284. States Exhibit 284 offered.
No objection. All right. Without objection, 284 is admitted.
And did your examination of Derek German's excuse me, let me back up. Were you able to develop a profile, a DNA profile, for Derek German from that standard?
Yes, sir.
And does that DNA profile give you any more information, in your opinion, on the hair identified as coming from an unknown female that came from the hand of Abby?
For the DNA profile, it was submitted for comparison to items that I had in this case, but given that I had already come to the determination that I believe that profile to be a relative of Libby, a comparison was completed with the profile of Derek German and, again, it just further supported my belief that the profile of the female individual was a relative of Libby.
And by examining Derek German's DNA profile, did it narrow the familial relationship that you believed was behind the identity of the unknown female?
It just showed some consistencies that you would expect among biological relatives that showed that the profile from the unknown is, not only could it be a relative of Libby, but it also appeared that it could be a relative of Derek German.
You also knew that Abby was found wearing Libby's sweatshirt. That information was provided to you, correct?
Yes, I was.
Did you share your conclusions that your examination of that unknown female profile and your examination of Libby's profile and her father Derek German's profile led you to believe that not only was it a female, but it was a female relative? Did you communicate that to investigators?
That was communicated just through investigative meetings and discussions that I had with the investigators. It was never in a report of any sort, as we don't make those types of conclusions without DNA standards from an individual. We don't include, I should say, those types of conclusions in our report.
And then, early this month, October 2024, I informed you that I was asking that you receive a STA dart of Kelsey German Siebert's DNA. Is that correct?
I think that you inquired about it even earlier than October. I believe we discussed it at the end of September. We talked about whether a DNA standard was submitted yet for the biological relatives.
Did you receive a standard from the sister of Libby, Kelsey German Siebert?
Yes, I did.
And were you able to develop a DNA profile?
I was.
And what was your conclusion regarding the unknown female on the hair on the right hand of Abby?
So the unknown female profile that was from one of the hairs on laboratory item 17, the hair from the right hand, was found to be consistent with Kelsey German Siebert and is estimated to occur once in more than 8 trillion unrelated individuals.
I'm gonna hand to you what's been marked or admitted into evidence as 1 States Exhibit 148. For your records, it's item 016. Can you tell the jury if you recognize that packaging?
Yes, I do recognize this packaging.
And did you receive that item at the same time that you received the items from the autopsy and from the swab of the tree and other swabs taken at the crime scene?
Yes, sir.
And what is it?
States Exhibit 148, which is my laboratory item 16, is the cartridge that I've examined in this case.
That came to you with all the other crime scene packaging?
Yes.
In this initial exam?
Yes, it was included as part of the original submission for this case. I received it on February 17, 2017, along with the other items I've examined and previously talked about.
And did you examine the contents of it?
I examined the item which was in this packaging, yes.
And did you document and note the condition it was in when you received it?
I did.
And what can you tell the jury about your documentation and what you know about the condition of that item?
Your Honor, I'd like to refer to my notes on my laptop, if that's possible.
You may.
Thank you.
So my notes indicate that I received the brown paper bag, which was sealed with clear packaging tape. Inside of that brown paper bag was a manila envelope, which was also signature sealed with clear packaging tape. In addition, there was loose debris present inside of the brown paper bag. Upon opening up the manila envelope, I observed one cartridge which had an imprint on it, Winchester .40 SNW, also contained loose debris, which appears to be plant material. I did not observe any visible ridge detail. The cartridge appeared slightly dirty. In my examination, I did not perform any serological analysis. I did take one swab of the entire surface of the cartridge. Upon taking that swab, I noted that the swab had a light brown, dirt-like staining present on it. Why'd you swab it?
So as part of my examination, I first did a visual exam to see if there's any kind of body fluid that warranted testing on that sample. There was nothing that warranted testing for a body fluid. So then I attempted to collect any potential skin cells that may have been on the surface of the cartridge. Swabbing. You
received this item amongst the other items received by you on February 17, directly from the hand of Sergeant Olihi?
Yes, I did.
In swabbing it, would you have, in doing that process with a moist swab, would you have removed dirt that was on it?
Yes. So part of the process is to wet tin a swab with a drop of water, so in doing that, yes, the swabbing would have removed the dirt that was present on that sample.
And you had dirt on the swab, or you had a substance that looked like dirt on the swab when you were finished?
Right, and it warranted me documenting that in my case notes, because I felt like, we haven't yet discussed this yet, but I feel like dirt could be impacting my ability to get a profile off of that sample, and that's why I noted its presence on the item.
I'm gonna hand you a box of gloves and a scissors. Ask that you open the outer packaging of State's Exhibit 148.
Do you care where I open it at?
I do not.
Okay.
You have a process you try to follow when you do this, is that right?
I do.
And this item's been opened before?
It has. So I am going to try tupanidina, on a different side than has.
For the record, would you describe it when you remove it from the bag?
I have removed the contents of the bag, which include a manila envelope that was the original manila envelope included in this packaging. I recognize, again, this envelope by my markings right here, which are not in the box here, but they do include the case number, item number, my initials, and my employee number. In addition, there is a piece of white paper that has been sealed across all of the edge. This paper I recognize again by my markings on the outside, as well as my initials across the seals on both edges.
Did you create that item there? Did you fold the paper?
Yes, sir.
And what's inside that?
This is what I referred to as exam paper. It is the paper that I performed my examination on. It is retained in items in which there might be loose trace evidence or debris that I want to preserve. So when I received this cartridge, there was loose debris, specifically appeared like plant material to me, and I wanted to retain that, so the paper is folded and sealed into a bindle, and each of the sides of the bindle are then appropriately sealed so nothing can come out of it.
I'm gonna ask that you remove the content of the envelope that was inside the bag in State's Exhibit 148, if you would, please. And what's the content of that envelope?
Within the envelope is a cartridge.
And is there, at the back, the head stamp of that cartridge? Are you able to see the back?
I can see the print on the end of the cartridge, and I can confirm by looking at my notes that this is consistent with what I've written down as part of my examination.
A Winchester .40 Smith & Wesson round.
I am no firearms examiner, but it does say a Winchester .40 SNW, which is exactly what I've written down in my notes.
And would you just hold that up for the jury, so they can see it? Did you mark it yourself?
No, sir.
And why's that?
At the time of my examination, there was still a pending examination, which means it needed to have additional analysis, so I did not place any markings on that item, as I didn't want to interfere with the additional analysis that needed performed.
But you were first in line to look at that in the laboratory?
Yes.
And you also gave it a look over for latent prints. Why did you do that?
Sergeant Olihi and I had discussed whether a latent print examination was necessary on this item. A lot of times with items such as these, we have our investigators make that consideration, and when we have something as small as this, it's nearly impossible to do both, a latent print exam and a DNA analysis exam, so we had that discussion and it was decided that DNA would be the preferred analysis for this item. Sorry, I can't remember what I was gonna say.
That's all right. But you could not see any latent prints visible, is that right?
That's where I was going. So I did just kind of visually examine the item to see if there was any kind of visible prints. Now, we know latent prints are not typically visible to the eye, but I wanted to note whether there was any type of ridge detail that I could observe. Sometimes if you have prints in like blood or something with grime or debris on it, that you might be able to kind of see a ridge detail. So even though we weren't doing a latent print exam, I wanted to note whether there was a print visible. If there would have been something that was present in kind of like a body fluid or a grease-like stain, then I would have then had further discussion with the investigator to determine if maybe we should have done that latent print exam.
What has been your success rate, and your DNA office's unit success rate with unspent cartridges and other ammunition?
So unspent and even spent cartridges are very difficult to get DNA profiles on, and it really just again depends on the quantity and quality of the sample, but you all saw how small that was when I held it up. My swabbing is designed to collect as many skin cells as possible. I need to have an abundance of skin cells present on your object to have enough DNA to develop a DNA profile, so we have found that, in our research in our laboratory, we have found that cartridges are not successful and, since 2017, we have removed them from analysis in our laboratory, for DNA.
They go on to either latent print and especially firearms/tool marks?
I believe that even latent print analysis has taken cartridges out of the scope of their examinations, as well, and again, it just goes back to the amount.
What was the result of your examination of your item 16, States Exhibit 148?
So I took a swab of that cartridge. I used that swab entirely, which means I consumed the sample entirely to maximize my opportunity of getting a DNA profile. The DNA result from that cartridge demonstrated an insufficient quantity of DNA for further analysis.
The jury has seen photographs of two Chuck Taylor Converse All-Star shoes, a right and left shoe. Did you examine two such items, and are they here, States Exhibit 146 and 147?
Yes, sir, I've examined these items, States Exhibits 146 and 147. They are laboratory items 14 and 15.
And items 177, identified as a sweatshirt with the word German printed on it, States Exhibit 177. Did you examine this?
Yes, sir, I also examined this item. I recognize it again by my markings.
And I'm gonna show you States Exhibit 178, identified as a shoe Maurice brand jeans.
States Exhibit 178 I also recognize as my laboratory item 30 and my markings, as well.
Would you tell the jury the results of your examination, beginning with the right and left Converse shoes and then the sweatshirt and the Maurice jeans?
So beginning with the shoes, as with all of these items I mentioned so far, I performed a serological examination. So I start with a visual exam and write down or indicate on my notes what I'm observing, what I see from the item. There was discoloration present on the tennis shoes that warranted me.
You've used the word inhibition and degradation. Would you describe those a little bit more for the jury and how they impact your ability as a DNA analyst?
Well, like I mentioned earlier, we have a part of our process can detect inhibition and degradation, so that was detected. So this result that I'm giving you is the result of that detection method, so it indicates these swabs of the shoes were inhibited and could not go on for further analysis. Essentially, inhibition is affecting the quality of the DNA profile, and degradation, as well. So I might be able to detect DNA, but I'm not going to be able to get a complete DNA result or even a suitable result that is suitable for interpretation because of the degradation and inhibition. I'm getting very, very limited information out of my DNA profile.
Do your notes reflect anything about the condition or what the source of the inhabitation may be on either shoe?
Yes. If I may refer?
Sure.
So item 14 and item 15, I have indicated that the shoe is heavy with soil and dirt and plant material. Yes, sorry, I wanted to read it all. It indicates that I was observing very heavy soil, dirt, and plant material on that shoe. To be honest, the shoes are very dirty, as, shoes can be, but these are in abundance. It wasn't just the sole or the bottom of the S-hoe, like the shoe, every part of the shoe. And given that they're Converse shoes, they were fabric, not leather, so that dirt was really embedded into that fabric material.
Would you describe what were your findings in relation to your examination of the sweatshirt?
I apologize, I haven't completed my results for item 15 yet.
The shoe?
Yes.
Which shoe are we talking about? The left shoe?
Item 15, which was the left shoe, yes.
Okay. Go ahead.
So I did mention that I did detect blood on one cutting of or on one sample of that shoe, so blood was detected in the tongue area of the shoe. The cutting of that shoe yielded a profile that was a mixture of two individuals. The major profile of that mixture was found to be consistent with Libby and estimated to occur in once in more than 8 trillion unrelated individuals. A minor profile was present, and that minor profile was consistent with Abby and estimated to occur once in 66 million unrelated individuals.
And that was on the tongue of the shoe?
Yes, sir.
Were there any other findings in relation to your items 14 and 15, the Converse shoes, that you wanted to share?
At this time, it's just worth mentioning that my visual exam included collecting hair, as well, and doing microscopic examination of hair. There was hair present, but it was found to be not consistent with human hair.
So your examination of the sweatshirt, that's an item that you looked at on multiple occasions, correct?
Yes, sir.
And in your initial exam on the March 7th report, could you summarize your results on the sweatshirt?
A visual examination was done of the sweatshirt. I will quickly note my visual appearances, but the black sweatshirt, I noted that their E was heavy.
And those were mixtures in which you were able to include Abby and Libby.
It was a mixture of two individuals, and both Abby and Libby were consistent with the DNA profile completely, so they appeared to be consistent with that mixture.
And was the hair found to be an insufficient quantity of DNA?
Yes, it was. And I also have results for those 3 swabs that I took, so the 2 cuffed areas and the swab of the front pocket. So only one of those swabs yielded an interpretable result, that was the swab of a cuffed area, and it was found to be consistent with Abby and estimated to occur once in more than 8 trillion. The other 2 swabs from the pocket and the other sleeve demonstrated the presence of a mixture in which the number of contributors could not be reasonably assumed or determined.
Thank you. Is there anything else you wanted to note to the jury about the examination of the sweatshirt?
Not at this time.
Did you examine these Maurice jeans that I believe were identified as taken from the body of Abby? Is that correct?
Yes, sir.
And would you summarize your findings for the jury of the jeans?
Again, I examined the jeans for the presence of any type of body fluids. Noted in my visual examination that there was heavy dirt-like staining present throughout the jeans.
Were you able to characterize when you say dirt? Did it have any appearance to you that you were able to note in terms of clay or sand or just dirt? Anything about that?
I didn't really make any conclusions. I'm not qualified to make conclusions as to what kind of dirt, but I did occasionally note whether it was a sand-like substance or mud. That's kind of as far as I got with my description on how that dirt appeared.
Did you have both on that?
Here I've indicated mud-dirt areas where Enhan said with my infrared light and my ultraviolet light, so that's what I was most likely observing. So in my serological examination, there was a lot of that dirt-like staining, and you can't really see underneath dirt to see what's there, so what I was doing was kind of testing for representative areas of the gene to see what might have been embedded under the dirt. So several areas were tested on the gene for the presence of blood and seminal material. There was no seminal material detected on the genes. Several areas indicated the possible presence of blood on the genes. The confirmatory testing was performed and it did not confirm the presence of blood. I ended up performing DNA analysis on 10 cuttings from the genes, in addition to 2 swabs. 1 swab was of the inside crotch area of the genes and 1 swab was from the button or zipper area of the genes. I also did a microscopic examination of the hair that was collected, and there was 1 hair collected from the genes that had a root, so DNA analysis was performed on that. Starting with that result, the hair failed to demonstrate a sufficient quantity of DNA. The 1 cutting of the genes yielded a profile that was consistent with Abby. A second cutting of the genes yielded a profile that was a mixture of 2 individuals. Libby and a laboratory individual could not be excluded as the possible contributors of that cutting, and it was estimated that 1 and 2 unrelated individuals could contribute to that mixture. The remaining samples either failed to.
Thank you. May we approach, Your Honor?
You may.
Sidebar conference conducted.
Just ready to move to the second and third.
Okay.
Exams.
Do we need to take a break?
I'd like to go move this evidence, so we can get the other set.
Okay. So this be a good time to take a break, then. Turn your microphone back on. It's off. You turned it off.
Judge, I don't know if you've been inaudible, and all points bulletin inaudible by the jury. Her vehicle was found in the area. It's unoccupied, and we believe she's in the court in the audience. I just thought I'd give you inaudible. We do believe she's the one that inaudible the jury and was attempting to photograph the jury.
Who is this?
Her name's Lana Oriani, R-I-N-I.
This is the...
She was the one in Fort Wayne, and she was in, so I just wanted to keep you in the loop.
Okay.
Inaudible, but I didn't want something to happen and me not tell you.
Is she inaudible, this woman? Or is she?
She's a YouTuber.
Oh, my God.
Inaudible, back in Fort Wayne police addressed it with her, and we put out her vehicle description.
Affirmative response.
We found her vehicle about 2 blocks from the courthouse. It was unoccupied. We think she's in the audience, but we're not 100% sure, so...
Do your people know what she looks like?
They do.
Okay.
They saw her walking inaudible, and so I don't know if Jason and Jeremy know inaudible.
So we can take a break and we'll discuss that.
Sidebar conference concluded.
All right. This is a good time for us to take a break.
All rise.
Jury out at 10:47 a.m.
You may be seated. So, Ms. Bozinovski, you can go ahead and step down from the stand while we take our break. You may need to also take a break. And those of you in the audience, if you just sit tight for a minute. Gentlemen, you may take Mr. Allen.
I didn't hear what you said.
I said, gentlemen, you may take Mr. Allen.
Oh, thank you.
Thank you. And let's take about 15 minutes. Jody, you can go off the record.
Off record at 10:49 a.m. On record at 11:09 a.m.
Okay. We are back on the record. I know we're waiting for Mr. McClelland and Mr. Luttrell. All right, Mr. Luttrell, have you switched out all of your evidence?
I have, Your Honor.
Okay. Are you ready to proceed?
As expeditiously as I can.
Okay. Let's bring them in.
All rise.
Jury in at 11:13 a.m.
Thank you. You may be seated.
Thank you, gentlemen. All right. You may proceed, Mr. Luttrell.
Thank you, Your Honor. And now to transition, ma'am, to your March 24th, 2017, report, and in that matter you looked at items removed from the body of Abby at the autopsy, 3 garments. Is that correct?
Yes, that included 3 garments.
And also items obtained at the crime scene and then a number of items removed from the creek. That's how they were described to you. Is that correct?
Yes, that is the description I received.
Let's look first at taken in those groups I have, for the record, States Exhibits 179, 180, and 181. These are identified as your lab numbers 31, 32, and 33. Is it okay if I give little Bulky you want to handle those 3?
Yes, sir.
The first item, your lab item 31, was a pink top, a sleeveless pink top from the body of Abby, and the second item is the gray cotton bra, your item 32, and the black bra is item 33. Would you explain to the jury, item by item, what your results were of your examination?
Yes, sir. These clothing items were examined for the presence of body fluids. Blood was detected on the pink sleeveless shirt from Abby and the gray.
When you're talking about a single source conclusion and you identify and express it that it's consistent with Abby, for example, then you add a number that says something like 8 trillion. Would you give that phrase again and explain why you word it that way?
So my report says the DNA profile obtained from the cuttings of the bra are consistent with Abby and are estimated to occur once in more than 8 trillion unrelated individuals. So this is a statistical calculation provided to give some weight or meaning behind the profile that I have determined from the sample, and it is, essentially, a stat that's showing the rarity of that profile. So it is my chance of selecting another unrelated person in the population that would be consistent or match that profile. So, essentially, I would not expect to find another person in the population to match that profile, or my chance of doing so would be 1 in more than 8 trillion unrelated individuals. So I often say if I had a room of 8 trillion individuals, there may be, one other person.
Thank you. I'm gonna hand you what's been marked as into evidence as such, and it's identified as a black Nike tennis shoe. You've already testified about examining the Converse shoe from Abby. Is this identified as an item found underneath the body?
Yes. This is a black Nike shoe, my laboratory item 39, that was identified to me as being recovered under the body of Abby.
And you examined it?
Yes, sir.
By the way, for the record, did you receive that item? Are you able to tell from your current documents you have opened from whom you received it?
Yes, sir.
And who is that?
I have received this item and the other items I just mentioned from Sergeant Brian O'Leahy through a evidence transaction that included another officer in my building, Lieutenant Berkey, who facilitated that evidence transaction.
What were the results of your examination of the Nike tennis shoe found below Abby's body?
This item, the same visual exam that I perform in all items to note the condition of the item, as well as to determine what type of testing needs to be performed. I determined that there was blood present on the shoe, so blood was detected. There were 7 swabs of the shoe collected. In addition, I collected 2 other swabs of the shoe. I also observed hair and did a microscopic examination for hair, but the hair did not have a root material.
Do shoes pose, in your experience, challenges or yield results that lay people may not understand or anticipate?
I mean, shoes have the same type of challenges that, and this shoe in particular, that all shoes have. They are covered in dirt. And dirt is, when you're swabbing it for potential skin cells, you're also collecting all that environment insults as well. And then also, shoes we wear them every day, we wear them into public areas, we wear them, in our homes, so they're a good item to collect a lot of DNA, as well.
Was this shoe, the Nike shoe, was it the same or different in terms of what you noted of the dirt or soil on it as it compared to the Converse shoes? Was it similar?
I'm gonna refer to my notes just to recall. Yes, so the shoe was in the same condition. I've noted that it has heavy soil and plant material present throughout the shoe.
Thank you. I'm gonna hand you now envelopes that are admitted into evidence as States Exhibit 158 through 167, noted as being various swabs from blood areas near and around the bodies of the victims. Would you, beginning with 158, or your item 40, would you mind telling us if you recognize those?
So you have handed me States Exhibits 158 through 167, my laboratory items 40 through 49. I have examined all of these items and do have results.
Beginning with States Exhibit 158 and your item 40, would you tell us what those were?
If I may, I will try grouping those items to the best of my ability. Beginning with a grouping of items 40 through items 43, items 46, and items 47, all of those are swabs from the ground or swabs from trees, and blood was detected on all of those items. All of the swabs were found to be consistent with the DNA profile of Libby and is estimated to occur once in more than 8 trillion unrelated individuals. Swabs of the ground, item 44 and item 45, blood was also detected, and those profiles were found to be consistent with Abby and is estimated to occur once in more than 8 trillion unrelated individuals. And finally, items 48 and 49 were swabs from the left index finger and left pinky finger of Libby, and serological testing was performed on those swabs and it indicated the possible presence of blood. I did not perform confirmatory testing on either swab to preserve it for DNA analysis. The results from both the swab of the left pinky finger and left index finger both were found to be consistent with Libby and occur more than once in more than 8 trillion unrelated individuals.
Is it a fair summary, an accurate summary, that of these swabs taken from the ground or the tree, 7 of them, the 2 you noted is from ground near between tree and Abby and their stain below the head of Abby were the only ones that 4 that had the DNA profile of Abby. Is that correct?
Yes, only those 2.
The items taken from the swabs of the finger of Libby and the left pinky of Libby, as well as swab from ground area of a blood area between marker 3 and 4, swab from ground at the feet of Libby, a swab from the ground below Libby shed, swabs of the ground near tree with blood on it, swab from tree with red stain, swab with red stain from tree below the previous item, those were all whose DNA.
All of the swabs that you've just mentioned were consistent with the DNA profile of Libby.
You were provided a number of items that were identified as collected by CSI Sergeant Jim Cody and designated as coming from the creek. Is that correct?
I don't recall who the items were collected by.
Okay. But they were identified as having come from the creek?
Yes, sir.
And I'll try to group them as easily as possible. But I'm gonna show you first States Exhibit 140. Ask if you can identify that.
States Exhibit 140 is a package that contains a sweatshirt that was recovered from the creek, my item 68.
141.
States Exhibit 141 is laboratory item 69, which is a pair of underwear recovered from the creek.
142.
142 is laboratory item 70, which is a sock that was recovered from the creek.
143.
States Exhibit 143 is laboratory item 71, which is also a sock.
144.
States Exhibit 144 is laboratory item 72, which is a green scarf, head scarf, that was recovered from the creek.
145.
States Exhibit 145 is laboratory item 78, which is a tank top shirt recovered from the creek.
Apologize for changing the sequencing here, but we're going to look at Exhibit 138, or, excuse me, States Exhibits 137, 138, and 139. I'll hand you first 137.
States Exhibit 137 is laboratory item 65, which is the other Nike shoe, which was recovered from the creek.
And Exhibit 138?
States Exhibit 138 is laboratory item 66, a pair of blue jeans, also indicated as recovered from the creek.
And Exhibit States Exhibit 139.
And States Exhibit 139 is laboratory item 67, which is a tie-dye shirt. Excuse me, also recovered from the creek.
Did the being found in the creek, did that pose any challenges for you as a DNA analyst?
I believe that it did. I believe that affected my overall results from the items that were recovered from that area.
Why do you believe that?
The creek, well, based off of my results, first of all. Like I mentioned, I am able to detect inhibition and degradation in my results, and so I have metrics.
Were you able to just go through your results, summarize it for the jury, and proceed in the order to identify the item that you're talking about, the order that's most convenient with you for your notes?
So we'll start with the Nike shoe, laboratory item 65. I examined that shoe, again it was a heavily soiled item, but I did attempt to collect any type of skin cells that may be present on that shoe. There was no blood, there was no seminal material. That type of testing was performed on the staining that was visible. There were 5 swabs collected from the shoe. One of the swabs was found to be consistent with Abby and estimated to occur once in more than 8 trillion unrelated individuals. 2 of the swabs gave me a partial DNA profile in which I couldn't determine the number of contributors, so I could not make any conclusions. An additional sample failed to demonstrate enough DNA. So overall, I was only able to obtain a DNA PRO file from 1 swab of that shoe. Moving to the jeans.
If I could just stop you there. This was a shoe unlike the shoes found on Abby, the Converses. This is a shoe that is the other half or matches, what you could tell, the shoe found beneath Abby. Is that correct?
That's correct.
Would you be surprised, given your finding, That it was identified as Libby's shoe?
Would I be surprised by the result?
Maybe that's a dumb question or a trick question, but can you answer it?
Well, I would just say that I can't really tell you how someone's DNA profile gets onto an item. That particular swab that I identified, the DNA profile of Abby was from the sides of the shoe, so the rubber sole on the bottom, that portion was swabbed and there. It yielded a single source profile consistent with Abby, so I can't make any conclusions on how she would have gotten there. I wouldn't find it unreasonable of a result, but I can't say how it got there.
Fair enough. Next item you wanted to discuss, was that which one?
Moving on to the genes again, heavy dirt, mud staining present. Plant material was present. I attempted to swab some areas of the genes, so there were 2 swabs of the genes collected for possible skin cells. Those swabs were again from the button and the zipper of the genes and from the inside crotch area of the genes. That sample from the inside of the genes did yield a mixture of 2 individuals. There was a major profile determined, and that major profile was consistent with Abby, estimated to occur once in more than 8 trillion unrelated individuals. There was also a minor profile. The minor profile was found to be consistent with Libby and estimated to occur once in 7.6 million unrelated individuals. And then the second swab from the zipper and the buttons showed me the presence of a mixture, but I could not determine number of contributors and, therefore, no further conclusions. And if I can elaborate on number of contributors, these mixtures were very partial in nature because of the inhibition and degradation present, so I had enough DNA present to develop a profile. I had a profile, but the profile was very, very limited information, and most of that information was falling below thresholds that we.
The next item?
The next item is the tie-dye shirt. The tie-dye shirt was just very heavily stained with mud. It had possible plant material, as well. I did test several areas within this shirt. Again, it's covered in mud, so I can't see. If there was a blood stain on it, I would not have been able to see it, so I was randomly testing and targeting areas within the mud. However, no blood was detected, no seminal material was detected. I did take one cutting of the sweatshirt, no blood was detected, no seminal material was detected. There was one hair with that shirt, but that hair failed to demonstrate a sufficient amount of DNA. The sweatshirt, which was a zip-up hooded sweatshirt.
What color? Did you note the color?
Yes. I noted that it was gray. It was very light in color. I noted gray. It could have been gray from dirt, but it looked gray to me. It appeared slightly dirty with slight plant material. It was cleaner than some of the other items recovered from the creek. I detected several areas on that sweatshirt that were enhanced with fluorescent lighting, so they fluoresced, but it didn't give the visual appearance of like a seminal material stain or a saliva stain. It was very streaky in nature, so it kind of appeared like maybe detergent or some kind of chemical stain, but it all tested negative for blood and seminal material, but
Did you cover your item 67, the tie-dyed shirt?
Yes, I did.
Did that complete your examination of items from the creek?
Of the items that
were submitted to you?
Yes, sir.
And did we go through your entire March 24th report?
I believe there's one additional sample. It's just a swab of a tree, item 86.
Okay. That was a tree that was identified as coming from where?
It says a swab of a tree below Rock Trail on east side of High Bridge. I think it was just a swab in which I think it's a swab which was just collected to see if anyone may have touched that tree. Regardless, I did not perform any serological testing, so it didn't warrant testing for blood or anything, and there wasn't any DNA present on that swab.
Thank you. We have talked about your examination of numerous items on March 7th and March that resulted in a report issued on March 7th and March 24th. In those, you did not find a profile of a male. You were not able to develop the DNA profile of any male. Is that correct?
From those 2 reports we talked about, I was not able to identify a male profile.
Your next report was April 11th. Is that correct?
Yes, sir.
And did that involve a re-examination by you of a black hooded sweatshirt recovered from the body of Abby?
Yes.
And did it also include an examination of your item 263, Indiana or States Exhibit 169, identified as a swab from the iPhone of Libby?
That report does contain my examination of laboratory item 263, which was identified to me as a swab of cell phone found under Abby.
I'm gonna hand to you. Is this the envelope that you received that you examined the contents of?
Yes, sir. I recognize this envelope, States Exhibit 169, as my item 263.
Let's start with that one first, and we'll come back to the sweatshirt. What was the result of your examination of the swab from the iPhone?
There were 2 swabs present in item 263. I did not perform any serological testing, which means there wasn't any blood-like staining present on that sample. I took those swabs directly to DNA analysis, and both of the swabs demonstrated the presence of a mixture in which the number of contributors could not reasonably be assumed, therefore, no conclusions were drawn.
On the black hoodie, what did you do on the second examination of this sweatshirt?
In the second examination, I went back to the sweatshirt and decided to take some additional samples of that sweatshirt. So initially, I had done what serological testing on the sweatshirt for the presence of blood and seminal material, and I had tested all those areas, and I had done 3 swabs from the sweatshirt initially, from the cuffs and the pockets of the sweatshirt, and I had chose those areas original. Why? Because those are, in my opinion, those are areas that may have come in contact with a suspect, so I was interested in seeing if I could get skin cells from those areas. Given that the sweatshirt was of importance, in that it was in the middle of the crime scene and the front of the sweatshirt was not as exposed to some of the environmental insults that the creek items were exposed to, I wanted to go back and do some maybe untraditional swabbing of that sweatshirt. So I went back and I swabbed the majority of the front of the sweatshirt and the sleeves, both front and back, of the black hooded sweatshirt to potentially collect any type of skin cells that may have been deposited within those areas.
Would you tell the jury the results of your analysis of those items?
I apologize in advance, I took 22 swabs from the sweatshirt. So in doing so, my attempt was to avoid the blood-like staining that I also knew to be present.
Let's go ahead. And were you gonna go on to discuss other items?
Yes, sir.
Let's go ahead and address that. You later did more analysis on that mixture, which you noted as 29A and B, black hoodie, your item 29D10. Did I have that designation and subitem correct?
That's correct.
29 is the your number for the sweatshirt, D10 is the subitem you created, which was a swab, and IT had the mixture of 3 people. What were you able to determine about this third person, the unknown male?
Well, I had an unknown male profile initially. Later on, I entered the unknown profile into our CODIS database system to be searched against any potential offender samples. Later on, I wanted to enhance that profile as much as possible, so at a much later date, I was able to use new technologies. So as we talked about earlier, the forensic community is always changing, and so I used a newer technology to help with interpretation of that sample. In doing so, I ran my unknown profile through an employee database for a second time and was able to get a match to that unknown profile to an employee sample, so therefore it was removed from the CODIS database and no longer searched because of that identification.
And that was that personnel, was that a lab employee?
It was a laboratory personnel person.
And your protocol is that you, and this is part of your QA/QC program, correct? That you do not identify that person.
Yes, sir. And just to elaborate, running our unknown profiles through an employee database is also part of our QA/QC profile. That was done initially, and it was done again a second time. It did not match to the employee database the first time because that employee was not in the database the first time, where they should have been, and it was not, so as part of our quality assurance/quality control program, a corrective action was placed.
Were there also changes made in terms of laboratory personnel and crime scene investigators sometime after 2017, in terms of their protocol to follow in terms of apparel to wear? Did that occur after sometime after 2017?
I can't say for certain, I'm not really involved in the training of our CSIs, but I do know that because of this particular instance, it was communicated as to what would be appropriate personal protective equipment to be used at a crime scene.
So encouraging the use of a mask in close proximity to this type of work, close proximity to certain items or for individuals, inaudible?
I cannot say what exactly was communicated, 'cause I really don't know. I was not privileged to that conversation. I just know that the conversation existed.
What proceed with the rest of your examination? Were you able to group any of the items sub-items?
I will speak in generalities, if that's permissible. The rest of the samples.
And other than the lab personnel, you found no male DNA on the sweatshirt?
I found male DNA on the sweatshirt.
I'm sorry. You could not develop a profile of the male DNA?
That's correct.
And in terms of the male DNA that you found on the sweatshirt, is there any more that you can say about that?
No, just that in that quantitation method that I mentioned earlier, I am able to detect the presence of male DNA, and most of those mixtures did, indeed, have the presence of male DNA present. I just did not determine an unknown male profile from any of those samples.
And again, we talked about clothing earlier and individuals living within a.
So clothing is, given the substrate that, of clothing, it's able to hold on to DNA. And then, when you launder DNA, as well, DNA can be deposited. So a lot of times, we think that cleaning and washing our clothes will remove the DNA. In fact, it's not. It's probably mixing the DNA from past washes in the current wash. So yeah, I often say that clothing has background DNA. So this morning, my husband gave me a hug. I have DNA from him. I've talked to Jim, I've talked to several other people today. So clothing can actually kind of hold on to DNA from everyday interactions.
I'm gonna ask you about a report that you generated that was dated July 11th, 2017, and asked, did that consist of examination by you and your results on looking at tree branches?
Yes, sir.
And would you tell the jury where you went to examine and begin your collection and analysis of the tree branches?
Yes. I examined a few items containing tree branches, and that examination was done at the Indiana State Police Lafayette Post, where those items were being stored.
Were they in a secure location in a building with other evidence items?
Yes, they were.
But this was an outbuilding of the post, is that correct?
That's how it was explained to me, yes.
What did you do with these branches? Explain to the jury.
Well, I essentially converted a conference room into an examination area, so I was able to provide for myself a clean examination surface with clean bench paper, and I brought forth some of my laboratory supplies. But I examined the tree branches essentially for the presence of any blood that may have been there, but I was primarily interested in collecting skin cells, or swabs for possible skin cells from those tree branches. So in my examination, I took several swabs from each of the branch for maybe how that tree branch may have been handled, and then I also collected some swabs for possible blood. I did not perform any of my serological testing at the Lafayette Post. I just took the swabs back to my laboratory and did my laboratory testing once I returned.
And the results of your examination?
So for grouping purposes, I took several swabs from each of the branches. So imagine, a lot of the branches were upwards of 7 feet long, so I partitioned that into sections, and then I also kept in mind, like, if you were to handle or move that branch, how would you have done so in my swabbing? And all of those swabs essentially failed to demonstrate a sufficient quantity of DNA, or they had that failed quantification test control, and that test control, again, is an indicator of inhibition, so I had several of the samples indicate that inhibition. There were 2 samples that I was able to get a DNA profile from the branches. One was from laboratory item 547, this is a swab of the branch which did test positive for blood. Excuse me, oh, there you go. I wanted to make sure, yes, it did test positive for blood, and that profile was found to be consistent with the profile of Libby. Then, a second swab which also tested positive for blood, which was my item 548 to 547 and 548, both of those items had A.
Did those branches pose unique problems for you with trying to swab and collect and then to be able to examine?
I mean, I can only imagine that those branches, being what they were as far as an evidentiary item, impacted my ability to get a profile. So the branches are covered in dirt. As I'm swabbing it, I'm picking up the plant material, the particulates from the dirt, and dirt is a known inhibitor. The humic acid within dirt is a known inhibitor to our process, so I do believe it played a role.
Ma'am, I'd like to conclude by going over some topics that relate overall to the work that you did, and you might draw our attention to some specific items in this examination that you've covered already. But you've talked about the inhibitors and degradation. Besides the branches, what other items come to mind that pose difficulty in you dealing with the inhibitors that were on and the degradation?
It's difficult to name items. I would say.
Let me ask you specifically, and then you can comment. What about the genes found on the body of Abby?
Do I think that I was dealing with inhibitors?
Yes.
Yes.
What about the clothes from the creek?
Yes. Like I mentioned earlier, these items were covered in dirt and mud and plant material. They were exposed to water and sunlight, and even though that may not have been over a long course of time, all of that plays a factor in my ability to recover DNA from these types of items.
What about the nature of given your experience, the nature of your item 16, states Exhibit 148, Winchester Smith & Wesson .40 cartridge? How would you characterize that difficulty? Do you know why the laboratory no longer accepts those to examine?
Yes. The item 16, the cartridge, again is a very small surface area. It's unusually minimally handled in general, so it's kind of like a one-time contact. It's not something that is frequently handled over and over and over. So I really haven't expressed that the success of skin cells, again, based on quantity and quality is based on frequency of use, how much something is handled, how much skin cells can be deposited. And for DNA to be successful, I need to minimally be able to detect, between 25 and 50 cells. And a surface area like a cartridge, being so small, that type of DNA, that level of DNA isn't typically deposited on such a small surface area.
Another topic I wanted to ask you about that we include is hair. What I think you've indicated that hair is transient. Explain what you mean by that, and what challenges would hair impose to you as a DNA analyst?
First, just the challenges of hair. Hair is typically naturally shed from our body, so in that naturally shedding process, the follicles and the skin cells kind of shrink. And when shrinking happens, and that's, what helps shed that hair. But when the shrinking happens, then the cell can kind of burst, the nuclei burst, which means the DNA starts degrading, and it happens quickly. So for hair to be successful, it has to kind of have like a skin tag present, and the best way to get skin present on a hair is through forcibly removing hair, and naturally shed hair isn't typically very successful. Overall, I can give you, just from that first report that I looked at, I examined like 80 hairs.
I don't want to get into the details, but just to quickly touch the surface of rootless DNA rootless hair technology, there are explorations underway to how to deal with hair that has no root or has no skin attached. And did you send in light of that? Did you send hairs to the FBI?
I did send a set of hairs to the FBI. It wasn't necessarily because of the rootless hair technology at the time, but the rootless hair technology has come to the forefront as a newer technology that's available for hairs in which there is no longer a root.
And did you ask or did the FBI provide you a screening service to help you focus on hairs that might be of some value?
They did provide a screening source to help identify hairs, and it's for a testing that wouldn't have been for me or my laboratory, as we had done everything that we could with the hairs. The only thing that hair can be and the only thing that our laboratory can do with hair is hair that has a root, and we would perform an autosomal STR technology, so we did send a set of hairs to the FBI to identify their laboratory to identify what hairs may have been not consistent with the victims, so that we could consider any type of future testing. And at the time, that testing was mitochondrial analysis, which again, is not performed in my laboratory. Since that time in 2018, when I sent the hair to the FBI, the rootless hair technology has kind of come into discussion.
And were these generally short hairs that you sent?
there actually was a variety of hairs sent. So anything that I had identified as a human hair, long or short, and even if I'd already identified a profile, except for the ones I already knew to be consistent with Abby, I went ahead and sent that to the FBI.
And what did what return to you as identified as unlike the victim's hairs? How many?
The FBI issued a report, and that report indicated to me that they found 3 hair.
Objection, hearsay. Any response?
I think as a DNA analyst and expert in this area, she's able to report what she learned from colleagues and consultants that she deals with. And I think this would be in that vein that she could go ahead and communicate the answer to the question I posed, which was how many hairs did they identify as unlike the victims. I think I wouldn't go any further, but I think that's appropriate for her, since she was relying upon that. But as an expert herself, Your Honor, I believe she would be able to do that. And the objection goes to the weight of the evidence, and not the admissibility.
Overruled.
How many hairs did they submit to you with that designation? Did you tell us already?
I did start to answer. Their laboratory report, which was addressed to me, did identify 3 hair that they found to be not consistent, just from a microscopic examination, to either Abby or Libby.
How many did you send them?
I sent them 72 hairs.
And what was the next step then, if you have those 3 hairs? Did you explore the possibility of rootless hair examination of those 3 hairs?
Yes. Our investigators inquired about what more could be done with those hairs, and that was explored as an option.
And what did you conclude after determining the options, and what the limitations and characteristics of that examination would be?
Well, I didn't really make any conclusions. So.
I guess what did you learn?
Yes. My role and the role of my supervisor, who's also the case manager of our laboratory at the time, was just to kind of research what technologies that were present in the forensic community and present that to our investigators so that they could make conclusions and choices on what type of testing may need to be performed, if any, on that hair.
What did you learn about the success rate of that technology?
So our research, and with consulting with several, actually not several, a few private companies that do that type of testing, showed that their success rate for rootless hair testing was very low and, overall, it might be better to not perform that testing at this time and wait for technologies to improve.
And what if testing it, what would happen to the sample? What happens to that if you test it?
Given the amount of hair that is needed for rootless hair testing and even mitochondrial analysis, it would most likely consume that hair in its entirety. There needs to be at least 5 centimeters of hair for that type of testing.
And given the low success rate, the consumption of the hair, and the hopes that technology would improve someday, what did you make a recommendation, not a conclusion?
I hate to say recommendation. We presented all of those findings from the just consultation with the private companies, cause this is a testing that's not performed in our laboratory, and we presented that to our contributing agency and investigators to let them make that decision, and a decision was made and I understood that decision and was in agreement with their decision.
You didn't make the decision, but you thought it was a reasonable one?
I think, given the low success rates and it was a one-time opportunity, it was a reasonable decision.
Also, the transient nature of hair?
There was a concern over what the 3 hairs that were identified and from what items they were identified on. If we were going to do newer technology and testing, it was a concern of maybe, I submitted 72 hairs, so maybe identifying which hairs were the best use of our resources, of the contributing agency's resources to do that testing. It just was. It's a very delicate balance at that point on were these 3 the most informative and meaningful hairs or were there others that would provide more meaning.
Last topic, touch DNA. You've done studies and read studies on that and have a practical experience in the laboratory in dealing with looking for skin cells on items. Is that correct?
Yes.
Has anyone ever come up to you and said, "Is my DNA there?"
Yes.
Does that ever happen?
All the time.
In court?
In court.
Okay. And what's the answer?
So first, I'd like to define a little bit of what touch DNA is, because a momentary contact, a physical touch, is different than examining for, skin cells from an item that is known to be handled and worn by someone. So touch DNA, yes, there may be DNA there, but it's quite possible there's not DNA there. It's dependent, again, on quantity and quality, how much DNA is in my hands, what the substrate is, the friction and the force that is applied to that single touch, the substrate, if there's any body fluids on my hands when I touch something. It all plays a factor, so I can't say for certain that, yes, I will get a profile.
Really, you're hearkening back to one of our earlier topics, which was good and bad sources of DNA, is that correct?
Yes.
Does the quantity of skin covered by clothing affect the number of skin cells that would be left behind, in your opinion?
So for skin cells to be deposited on something, that skin would need to be exposed. So if the skin was not exposed by being covered by clothing, then it could affect it, but it also, again, depends on what type of DNA is present on the clothing item itself, as well.
If gloves are worn, obviously, that would affect touch DNA involving the fingers and hands.
It would prevent the transfer of the DNA from the fingers and hands, but mind you, there could also be DNA on the outside of the gloves, but yes, it would prevent maybe the shedding from that direct contact.
Do you often have scenarios in your work, your laboratory, of examining clothing items left behind by a perpetrator, a ball cap, a shirt, or something that might be left behind, and so is that something that you've done in the past?
Yes. So clothing items that are left behind are kind of typical, routine evidence for our laboratory.
Weapons left behind, do you examine those for touch DNA or skin cells?
Yeah, that's something that, if it's something is left at a scene or dropped, that is a normal type of item that we may examine.
Would you tell the jury the difference, in your after your research and your training, the difference between handling an item and touching an item?
It really just goes back to the amount of DNA that's being deposited, so an item that is being worn or handled has, is greater frequency of contact between the skin and that handling and wearing of an item, so a greater chance of skin cells being deposited. So we don't necessarily consider clothing that we're examining for potential wear as a touch DNA sample, like we don't define those as one and the same. So touch is more of like just a, very limited contact with an item, not something that we persistently had contact with through use or wearing.
And if it sounds then like you're saying the frequency of use is an important factor in whether or not you can find touch DNA?
Yes. There are a variety of factors that play a role in how much DNA I can recover from an item.
Is it understood in your discipline the notion of shedder versus non-shedder when it comes to skin cells?
That's a terminology that's commonly used in studies in our discipline. We don't typically use that in our analysis, but yes, I am familiar with that terminology.
And what does it mean?
It, basically, just means that studies have shown that some people can naturally shed more DNA than others, and that might not be like all the time. It might just be specific to a day or a time or even season, but some people have the capability of shedding more DNA than others. We do all shed DNA, it's just who's the good shedder and a poor shedder, low shedder/high shedder.
As a general rule, can the skin cells on the skin that are subject to coming off, can they diminish with frequency of touch over a short period of time? Is that something that's been documented? Do you understand my question?
There have been studies, I do understand, thank you. There have been studies to show, like, if I touch something 5 times in a row, am I going to get more DNA the first time? The last time? There are studies of that, so I am familiar with that type of study. Again, it's just dependent on a lot of factors, force, friction, what's on your hands to begin with.
The competitive nature of good and poor DNA sources. Would you explain what that means?
I use that terminology a lot when I'm trying to describe my results in this case. Blood is a good source of DNA. I have a lot of blood off of the clothing items, detected a lot of blood off of the clothing items, item 29, item 30. And if the
killer were to touch the shoulders or the neck of either Abby or Libby in the process of this crime, how would the competitive nature of DNA, good and bad DNA, affect your ability to see if that touching ever occurred?
I'm just gonna say that trying to detect a touch sample in an overwhelming amount of blood is nearly impossible. There's just not gonna be enough deposit from a single touch unless that touch also has body fluids on it for me to be able to pull out just the DNA profile of just that skin cell component. The blood component is typically going to be.
Your Honor, I'm going to object. She's testifying on facts not in the record as to where someone may or may not have touched one of the victims or when they may or may not have touched one of the victims.
I guess it could be seen in the form of a hypothetical question, but I said if. I did not suggest that the record shows that at all.
Sustained.
Did you find the DNA of Richard Allen on any of the items that were submitted to you and described by you today that you examined?
No, I did not.
Were you able to develop a DNA profile of any male on any of these items?
I did not develop a male profile that was suitable for comparison purposes from any of the items that I've examined and mentioned today, excuse me.
Thank you. Your Honor, I have no further questions.
As we discussed, we're gonna go ahead and break for lunch. So, ladies and gentlemen, again, while you're outside the courtroom, please keep your minds open. Please don't form any opinions or conclusions about anything. You may discuss the evidence that you've heard so far, but please, only do that if all 15 of you are present in the jury room at the same time, and please, don't deliberate. Gentlemen.
All rise.
Jury out at 12:34 p.m.
You may be seated. And again, if you'll just kind of wait a little bit until the jury gets to the jury room. And I know it's getting warm in here, and it's been a long morning, but I've noticed an awful lot, well, several members of our audience sleeping. I don't conduct court in your bedrooms, and I would appreciate you not sleeping in my courtroom.
Judge, may we approach?
Yes.
Sidebar conference conducted.
The exhibit. I'll bring a copy of it.
Oh, okay.
Yeah.
Okay. Nick.
I talked with the clerk over here, and she was fine with me making copies.
I don't think I'm seeing her in here.
I've got a picture of her.
Okay.
I got a picture of her, and I'll give it to you so your staff will know.
Okay.
I didn't have their cell phones to be able to do it in morning court. I was trying to pay attention, so I just thought when we had our stopper here.
I shared all of that information from you with my staff. I'm not really keen on it and I don't know if you guys are, but I don't particularly relish the idea of them getting photographs or following you guys, so I don't know what this woman is about.
They already have mine all over, but...
Yeah, that's okay.
Well, I get it, but you know you're a public official.
Yeah.
Inaudible.
Sidebar conference concluded.
All right. Let's resume court at 1:30 then. And folks, you can go ahead and leave.
Off record at 12:37 p.m. On record at 1:37 p.m.
All right. We are back on the record. Ms. Auger, are you ready to proceed on cross? I am, Your Honor. Let's bring the jury in.
All rise.
Jury in at 1:41 p.m.
Thank you. You may be seated.
Thank you, gentlemen. Ms. Auger, you may proceed. Thank you, Your Honor. Ms. Bozinovski, you referenced, at the end of your examination by Mr. Luttrell, that there's some new emerging technologies and advancements in DNA.
Yes. DNA field is ever advancing, so there's always something new on the horizon.
What are those?
The new technologies that I was referencing with Mr. Luttrell was the rootless hair technologies that we're becoming more aware of in the forensic community.
And what are those new technologies as it relates to hair?
It's just a new extraction method that's I guess essentially been shown to be able to get autosomal DNA from the shaft of the hair.
What is that extraction method?
It's, as far as I know, a proprietary method. It's being used by private.
And there have been criminal cases that have been resolved by the use of SNP DNA, right?
Are criminal cases being solved by SNPs? Well, SNP or SNP analysis is typically used in sequence, the sequencing analysis. It's typically used in forensic investigative genetic genealogy. It can also be used on low and degraded samples, as well.
And people have used, and I said SNP, but you refer to it as SNP, right?
Yes, that's how I call it.
Okay. And you can obtain SNP DNA from hair shafts, right?
If you have an appropriate extraction method, then it's possible.
And that's happened in the past in other criminal cases, right?
I don't know of a case that's been specifically used on hair for SNP analysis.
Okay. And you are currently in the process of validating a lab at the Indiana State Police to improve your ability to conduct SNP analysis, right? And extraction?
Not validating a lab. We're validating an analytical procedure using SNP analysis.
So that you can increase your use of that.
It is.
Right?
It's a new tool that we can add to our technologies to potentially use on cases, yes.
Okay. Because SNP DNA provides accurate and reliable DNA extractions that can be used to solve crimes?
It's possible. Again, it depends what we're talking about, but we do currently perform SNP analysis in our laboratory with next-generation sequencing, and we do that right now only on unidentified and human remains cases, so it's not necessarily a suitable technology to be used on hair or mixture-type samples. It's not within the scope of how we use that analysis.
How you use it at the Indiana State Police Lab?
That's correct.
All right. And is that part of the reason why you're seeking to expand your ability?
The expansion of that new tool, which is to be used for forensic investigative genetic genealogy, is SNP analysis to be used for genealogy. Again, it's only applicable to single-source type samples that we can try to enhance those samples. So SNP analysis can't be used on a number of samples, but
it can be used on hair.
If that sample is single source.
Yes or no, can it be used on hair?
Your Honor, she's answering the question and...
Sustained. Let her answer the question.
If that sample is single source and it meets whatever quantification threshold that we determine with our valid DAT ion, then it potentially could be used. But, like I mentioned, we're still validating that analytical method at this time.
There are other labs that do that, right?
That use SNP analysis for hair?
Yes.
Like I said, I don't know of what other laboratories are using that right now. They're private companies that can do that, but I can't speak of a specific case that's been used.
Do you recall Mr. Baldwin and I coming to your lab this summer and speaking with you?
Yes.
And do you recall us discussing the use of forensic genealogy?
We did discuss the use of that.
And, in fact, you have cases resolved through forensic genealogy, correct?
Yes, we have.
All right. And you're in the process of bringing that technology in-house to your lab, correct?
We are validating it, and if it is a successful validation, then that is a platform that could be brought into our laboratory.
So that your equipment right now only looks at 200 SNP loci, right?
Our sequencing analysis that we do now for SNPs, yes, looks at approximately 200 loci for our unidentified and missing persons cases.
But in order to properly use it or to expand your lab, people generally look at 10,000 plus loci, right?
The new chemistry that we are evaluating is looking at more than 10,000 SNP loci.
All right. And by combining techniques originally developed for ancient.
Can you repeat that?
Sure. By combining techniques that were originally developed for ancient DNA, people have been able to generate DNA profiles from SNPs that have high level of discrimination between individuals.
That sounds like you're referring to the type of extraction protocol that I'm mentioning that was specifically designed for ancient DNA, and it's a proprietary extraction method. I don't have knowledge of that method.
Okay. Now, you kept hairs that you were not able to develop enough DNA to test, correct?
If the hair was not consumed in its entirety, then yes, there is hair shaft remaining from those hairs.
True or false? You kept hairs that do not have enough DNA for comparison currently? Is that true or false?
It's a difficult question to answer, ma'am. If I consumed that hair because it was already so small, then I do not have that hair any longer.
Do you still have any?
I do.
Okay. How many?
I know that I sent 72 hair to the FBI, so I imagine somewhere in that ballpark.
And you said you were holding on to those because there are advancements in DNA and you didn't want to risk consuming those by utilizing technologies that may or may not reveal results. Is that right?
No.
No? Okay.
Because I didn't make that decision. It's not what I necessarily wanted to do. Again, I consulted with some companies on what options they had for testing and provided that information to my investigators. That decision was made by the investigators and it is being retained in our laboratory at this time, any hair shafts, for potential any future testing.
What companies did you consult?
I personally didn't do the consultation, my supervisor did, and she was the case manager at the time, but we consulted with I believe Bode Technology and DNA Labs International.
For what purpose?
Those two companies were known to have an extraction process for rootless hair testing.
And were they able to test any of the hairs that were collected from the scene?
Like I mentioned, that decision was made by the investigator to not pursue that testing right now.
That wasn't my question. Are they able to test the hairs that you had that were collected from the crime scene?
I apologize, I thought you said did they test the hairs? Are they able? Yes, they could test those hairs if that was the decision made.
And the decision was made not to, correct? By law enforcement.
I believe the decision was made not to at this time.
At this time. But this is the time that Richard Allen's on trial, right?
Is Richard Allen on trial? Yes.
And so that's evidence that is available potentially to the state by outside labs that we don't have available, the jury doesn't have available today to use in their determination, is that correct?
Again, I can't speak to what information would have been yielded, if any, from that testing.
Because it wasn't done, right?
That testing has not been completed.
Has it been started?
I think I've already answered that question. It's not been completed or started.
Okay. All right. Now, you said you sent 72 hairs to the FBI lab, correct?
Yes, ma'am.
All right. And they sent you one report back dated May 20th, 2020, is that correct?
That's correct.
And they identified 36 items, correct?
It looks like there's 36 items on their list.
Thank you. And they also told you that if microscopic hair comparisons are requested, please submit known head samples from any subjects. Recall that? Top of page 3.
I do recall that.
Okay. And have any samples from Richard Allen been submitted to the FBI?
No.
Have any samples from anyone been submitted to the FBI?
No, they have not.
And those are microscopic examinations, correct?
Objection to what those are. I don't know if she's referring back to the hair examinations by the private companies or if she's referring to some other.
Clarify your question. Oh, sure. Yeah. The FBI examinations, those are microscopic examinations, right?
In their report, they've said if microscopic hair comparisons are requested to these hairs, please submit head hair samples.
So is that yes?
Yes.
All right. And microscopically examining hair doesn't use up the hair, right? You can examine it over and over and over again, correct?
I do not perform this analysis, but I believe that you are correct. that they would just do a comparison of the hairs.
So you could submit any number of hairs and still have the original hairs that you're comparing it to for analysis for later dates, right?
Yes, it sounds like it.
And at the state police lab, you only conduct the tests that law enforcement requests, is that correct?
The law enforcement agency is our customer, so yes, we perform tests on their behalf.
Well, do you perform tests that they do not request?
If the evidence warrants it. I guess I would need a more specific example.
Well, would if you become aware of a test that could be conducted, do you go ahead and do you call law enforcement and say, hey, this is available? What would you like me to do?
Again, it really just depends on what type of testing we're talking about, but if it's something that's like as simple as they asked me to look for blood, but I see something that may be consistent with seminal material, then I don't go and seek consultation with the investigator to test it for seminal material.
What about DNA comparisons?
What about?
Do you ask to compare the items that you have to known samples, or do you go ahead and compare them to the known samples that you have?
Well, typically, known samples are submitted for that comparison purpose.
Okay. Well, let's talk about item 17, all right? That is a hair recovered from Abby's right hand, correct?
I just want to clarify. Yes, from the right hand.
And you knew pretty quickly that it was a female hair, correct?
It was a profile consistent with a female individual.
And that it might be a female related to Libby. You also KNEW that right away, right?
Yes, there were genetic consistencies that made me think that it could be a female related to Libby.
And you had DNA samples from Becky Patty, her grandmother, correct?
At that time, I did not.
When did you get it?
I received the DNA standard from Becky Patty at the same time I received the standard from Kelsey German Siebert.
When was that?
That as recently. I'd have to refer to my chain of custody. I believe it was, I would hate to misspeak, but it was recently.
About 2 weeks ago?
Yes, somewhere around there.
All right. So as of March 7th, you had a hair from March 7th. 2017, you had a hair that you identified as unknown female, one that might be a female relative of Libby's, and no one asked you to compare it to Becky Patty or Kelsey German until 7.5 years later, after the trial started. Is that right?
Essentially, yes. My examination was mostly focused, like I've explained, on looking for male DNA.
Why?
There was indication that our perpetrator was a male individual, and that's what I was focused on, finding samples in which a male profile could be determined.
You had unknown female 1, which might be related. You also had unknown female 3, right?
There was another unknown female number 3 on a different item.
Right. And you had the hair from the sheet of the body bag in which Libby's body was sent to the 4 autopsy, is that right? Item 35, C.
I do believe that's where the unknown female number 3 profile was coming from, was a sheet from the body.
Thank you.
If I could refer to my notes though to confirm. Yes, ma'am.
Okay. So 35, C, was hair from the sheet in Libby's body bag, correct?
That's correct.
And that was a female hair?
It was consistent with a female profile.
Thank you. I always like to make that next leap. Consistent with a female profile. And so we have unknown female 1 and unknown female 3, which is item 35, C, right?
It was 35, B, 3, I believe. Unknown female number 3 was from item 35, B, 3.
Thank you. So the only DNA profiles you have from items that came from the crime scene were unknown female 1, unknown female 3, and male DNA that you believe is from one of your lab personnel, is that right?
I think that summarizes the unknowns that I developed, yes.
And yet, you were only looking for males, is that right?
I can't speak on behalf of the investigators, what they were looking for, but like I've mentioned, I screened several of the intimate samples for the presence of male DNA, and I'm at the discretion of whatever the investigators submit for comparisons to the unknown profiles that I've developed. I don't tell them how to do their jobs.
Okay. So they were telling you, hey, we're looking for male DNA? Were they telling you they were looking for male DNA?
That was just case information that I was looking, that we had a male perpetrator.
So that was what you decided, or law enforcement?
I followed the procedures that we have in the laboratory for a screening of sexual assault kits and intimate samples when there is a male perpetrator.
Well, ma'am, you don't know whether the perpetrator is male or female, right?
I had reason to believe, from the case information that I was provided, that it was a male perpetrator, so I screened the evidence collection kits looking for male DNA. And then, the other remaining samples from clothing and swabs, that type of screening mechanism was not used. Those samples were amplified and profiles were determined if there was enough DNA present, regardless if it was male or not. Just the intimate samples is where I was looking for male DNA.
Well, and you also said you were looking for male DNA on the black sweatshirt, right?
Yeah, I think that's what I just mentioned, regardless if it was.
Well, I thought.
Human or male, I was trying to find enough amplifiable DNA off of all of the evidentiary items with the exception of the intimate samples, which included the sexual assault kits and the body swabs. Those are the only ones that I had stopping point decisions made on whether there was male DNA or not.
Okay. Well, the sexual assault kits that you examined, those had nothing relating to Richard Allen, correct?
I did not develop a DNA profile from an unknown individual from any of the sexual assault kit items.
And so that could lead us to believe that neither Abby or Libby was sexually assaulted, correct?
I can't make those kinds of conclusions from my results.
But you have no evidence that they were, right?
I only can talk to the results that I have, which indicate that there was no male DNA present on the majority of the samples within that kit, minus the external genital swabs and fingernail scrapings, which just had an insufficient amount of DNA.
Now, how do you obtain DNA for comparison purposes?
The contributing agency submits standards for me to use for comparison purposes, so whatever they submit then I would compare to the unknown profiles in the case.
And how many standards do you think you received in this case? DNA samples?
I received hundreds of DNA standards.
And other than Libby and Abby, their female family members, and female law enforcement, how many DNA standards did you receive for females?
I believe one.
Out of hundreds? Out of hundreds?
Oh, sorry. Yes, one. I believe one individual was a female individual and, maybe two, but one.
And multiple people could have been involved in the murders, correct?
I cannot make any conclusions about what happened. That's not in the scope of my analysis.
Okay. Well, you looked through and you had some case information available to you, right?
I only had the information provided by the investigator at the time of.
And that is some information about the case, right? Do you think it's information about the case?
Right.
All right. And you yourself testified earlier that there were many items, many swabs, that contained mixtures of individuals, correct?
Many of my results were mixtures.
Okay. And some of those you can't limit to 2 people, correct?
There were a few mixtures where I was able to say at least 2 or at least 3 people, but I did not draw any conclusions from those samples.
I understand, but, in fact, I believe with the sweatshirt you said 3, maybe 4 or more, right?
I made that reference in my determination of number of contributors, that sometimes the information of such limited quantity that it's difficult to determine whether it's 3 or 4 individuals to give an exact number to a mixture.
Which means you can't limit it to 3, right?
Excuse me? Limit what? I don't understand the question.
Sustained. Which means you can't limit your analysis to 3 people, Libby and Abby, and only 1 perpetrator, correct?
In the majority of my samples, I've have results that say I cannot determine the number of contributors, so it's not even that it's 3, I could not determine the number of contributors.
Sure. It could be 3, 4, 5, 6.
I guess I would need a specific example of what you're referring to, because, like I said, the majority of my results say I cannot determine the number of contributors.
Meaning it can be more than 3, right?
It could be 1. It could be 2. But there was not enough information for me to determine the number of contributors.
I understand. Could it be more than 3?
Objection. That's been asked and answered twice, I think.
Sustained. I asked her if it could be more than 3 and she said it could be 1 and it could be 2, but she did not say whether it could be more than 3. You've made your point. Sustained. Move on. Thank you. Your lab takes extra precautions to avoid contamination?
We do have procedures in place as part of our quality assurance/quality control program that help limit contamination on evidentiary items.
And yet you believe that, despite those precautions, that the male DNA profile you developed was from one of your unnamed employees, is that correct?
Yes, there was consistencies in the male profiles that I developed between them and laboratory personnel member.
How many laboratory personnel have access to the sweatshirt?
I was the only laboratory personnel that was examining the sweatshirt, if that's your question, but that sweatshirt passes through the hands.
So can you answer the question? How many people in the lab have access to the sweatshirt?
I guess I don't understand the question. The packaging of the sweatshirt or the actual sweatshirt?
The actual sweatshirt, where you found the male DNA.
I can't put a number to it. I was the only person to actually examine that sweatshirt, if that's the question. It was, obviously, collected by somebody. I believe it was transported between posts and districts into the laboratory, but I don't have an exact number. I may be able to determine that from the chain of custody, but I don't know what happens to that item outside of the laboratory and outside of my chain of custody either.
Sure. And I'm not asking you about outside the lab, because it wasn't someone from state police whose DNA showed up on the sweatshirt, right? It was someone from your lab.
Well, I am with the state police.
Okay.
And?
Well, not someone from the post, correct?
The state police officers are considered part of our laboratory personnel employee database, as well.
So the male DNA you found on the sweatshirt could have been from a state trooper?
Could it have been?
Yes.
If they were in our employee database, then they would be considered, yes, as part of our employee database, a laboratory personnel member if they are involved in the handling and collection of that item.
Now, it's not, so despite your precaution, someone from the lab, their male DNA was on the sweatshirt, so it's not unexpected that DNA can be transferred from one person to a sweatshirt, correct?
It's not unexpected for DNA to be transferred between, obviously, people and the item.
Okay. And especially if you're handling a sweatshirt to the point that you're dressing a body with it, right? That's a lot of interaction from someone in that sweatshirt, would you agree?
Are you asking me a hypothetical situation of if it's possible?
There's no evidence of anyone dressing a body, Your Honor. I would object to the question. Facts not in evidence.
Sustained. If someone were dressing a body with the sweatshirt, would that be a lot of interaction that they had with the sweatshirt?
It would be maybe more than just a temporary contact DNA like we were speaking of earlier, but I hesitate to say whether it'd be enough to put DNA on an item, because there's a lot of factors in place, like what kind of personal protective equipment were they wearing and what was happening during a dressing event.
Well, did anyone at your lab actually touch the sweatshirt outside the packaging other than you?
I can only speak to what happened to that sweatshirt while it was in my custody, and no one other than myself touched it while it was in my custody.
Did you take the cuttings from the sweatshirt, or did you have someone else?
I did that.
And was there any other testing conducted on the sweatshirt other than what you did?
I am not aware of any. If there was other testing done, I'm not aware of it.
Do you have any male assistants assisting you?
During my examination, no.
Now, you've also reviewed and examined items that were collected from Richard Allen's home, is that correct?
Yes, there were items submitted that were indicate D to me as being collected from Richard Allen's home.
And you examined a number of items that came from his home?
Yes, ma'am.
And on any of those items that you examined, did you find any connection to Abby or Libby?
No, I did not find any DNA profiles from any of those items that were consistent with Abby or Libby.
And you examined a coat, a blue Carhartt coat?
Do you have a date on that report?
I believe it's November 2022.
Yes, I did examine a blue Carhartt coat.
And you examined a cutting from a carpeted area of Richard Allen's Ford.
Yes, ma'am.
You examined some dirty black boots?
That's correct.
Some knives?
Yes, ma'am.
And no evidence anywhere of DNA of Libby or Abby?
That's correct.
Now, you testified it could be difficult to get DNA from unspent cartridges, recall that?
Yes, ma'am.
And you said that was because you need an abundance of skin cells?
Well, I didn't say that was because of that. There could be a number of factors, but it could include the amount of DNA that would have been left behind on an item like that.
But you don't know how many times someone touched or interacted with that unspent shell, correct?
Right, which is why it's necessary to take that swabbing to attempt the DNA analysis on the sample.
And you can get DNA from metal surfaces, correct?
You can.
When you examined the cartridge, did it appear to have been impacted in dirt at any point?
I would not be able to draw that kind of conclusion from my examination of the cartridge. Like I mentioned, it had a slight dirt-like staining.
Okay. Slight dirt, is that right?
Yes, I believe that's.
And despite an abundance of blood at the crime scene, there was no DNA on that casing, that cartridge, from Abby or Libby, correct?
There was not enough DNA on the cartridge for me to develop a DNA profile from anybody, to be compared to anybody.
How many hours did you spend on this case?
I don't know. I can't put a number to that. As you can see, I've issued a lot of reports, I've done a lot of items.
How many items do you think you examined?
Hundreds of evidentiary items, along with the hundreds of standards that I've looked at.
Is it fair to say you put a lot of time and work and energy in this case?
Yeah, I would say so.
And nothing can be connected, nothing biologically, or I'm not gonna be able to pronounce this, from a serological standpoint connects Richard Allen to Libby or Abby, correct? Despite all those hours and time and energy.
There were no DNA results where I have an unknown male profile or a profile that's consistent with Richard Allen from all of the examinations that I've done.
Can you obtain DNA from spit?
Spit is, essentially, saliva, so yes, it's possible that would be a source of DNA.
Thank you. Mr. Luttrell.
Yes. Yes. So no DNA of profile consistent with Richard Allen or with any unknown male. Is that correct in your analysis here?
That's correct. I did not develop a DNA profile of any unknown male individuals from the items that I've examined and talked about here today.
These new ventures of DNA that Indiana State Police is entering and attempting to validate, you're a part of at least 2 of those initiatives, correct?
That is correct.
And would either one be of assistance to the jury in this case, in your view?
Like I mentioned, the SNP analysis that we're currently using in our laboratory that I am an analyst using for casework is only being used on unidentified human remains and missing persons casework and only for samples that are reasonable to be single-source profiles, so not mixture samples. It's not a suitable technology at this time for mixtures. The other method, which we're currently still validating, is something that may be used on forensic samples like hair, but that validation is still undergoing and would still warrant us having an appropriate extraction method to obtain DNA from the shaft of a hair.
Speaking of hair, The FBI indicated they did microscopic examination of hair, and they could do more, they said, and if you do, they had some instructions on how to submit it. Is that correct?
There was just a reference of additional microscopic hair examinations or comparisons could be performed upon submission of additional known hair samples.
And microscopic hair examination is no longer a legitimate form of identification.
Objection, leading. Sustained.
I'll rephrase it. Is it a legitimate form of identification in comparison, in identifying an individual?
It's not my specialty, and this is not a type of analysis that we perform in our laboratory, so we do not do hair comparisons in our laboratory to show whether hairs are consistent with specific individuals. That is something that the FBI is comfortable doing, and I believe that they acknowledge that as just a kind of an investigative tool, not an identification method.
It's a screening device?
Yes.
And the Indiana State Police used to have a hair comparison team or unit in the laboratory, didn't they?
Objection, leading. Overruled.
Our laboratory did used to do those types of comparisons way before my time. I'm not familiar with that, and it's nothing that's been in place in the 18 years that I've been with the laboratory.
Are you aware of whether or not microscopic hair comparison for? Just one moment, Your Honor. I think I'm about finished. Q. Was there any resource within the Indiana State Police DNA laboratory that you believed would be helpful that you did not employ here on this in your analysis and investigation? A. In my review of the evidence for this case, I feel like I have exhausted a lot of the resources that we have, if not all of the resources that we have in our. Thank you. I have no further questions.
Ms. Auger. There are other labs with greater capabilities than yours, correct?
I would hate to say greater. Each laboratory has different procedures and different processes in place for DNA analysis.
There are labs that can do things that your lab can't?
Yes, ma'am.
Okay. And you sent off cigarette butts for forensic genealogy, correct?
Yes, I did facilitate sending off those samples.
Because you didn't have the ability to do that at the Indiana State Police lab, right?
That's correct.
And would you agree with the idea that hairs without roots can provide mitochondrial DNA, which can be useful for genealogy purposes?
Yes, hairs without roots could be used for, or could have mitochondrial DNA performed on them. I'm not sure if the mitochondrial results can be used in genetic genealogy other than maybe assigning like a maternal haplotype group, but again, I'm not a mitochondrial DNA analyst at this time.
Are you aware that it can provide information on maternal lineage?
Yes, ma'am. That's the maternal haplotype that I was referencing.
And it can also be used to predict externally visible characteristics, such as eye, hair, and skin color.
That type of external visible characteristics is not coming from mitochondrial DNA, so those types of conclusions are based off of SNP analysis or SNP markers.
Okay. And you can get the SNP markers from the hair shaft?
Yes, through that.
Okay. Thank you.
Analysis.
Now, you sent hair, 72 or the 36 they received, hairs off to the FBI lab for microscopic comparison.
We're outside the scope of redirect, Your Honor. We've been over this. I would object.
No. He asked her about microscopic hair comparisons is well discounted.
I did.
Yeah. And now you're talking specifics, is that what you're doing again? Well, answer my question so I can rule on the objection. Yes. Overruled. Thank you. Sorry, didn't mean to hinder the process, so I apologize. You sent 72 or 36 hairs off the FBI for a microscopic hair comparison, correct?
Yes.
They didn't ask for it, you sent it and asked them to do it, right?
Who didn't ask for it?
The FBI didn't ask to receive the hairs, You sent them to them and asked them to test the hairs.
Oh, yes, sorry, I wanted to clarify.
Sure.
I sent that hair on behalf of my investigators, who did ask for it, so that's why I wanted to clarify. The FBI did not request those hairs, yes.
Okay. And that was and they responded in 2020, right?
The report is dated from 2020.
Thank you. Nothing further. Does anybody on the jury have any questions for Ms. Bozinovski? Would counsel approach?
Sidebar conference conducted.
What type of quality control is necessary when testing for DNA reagents? For DNA what? Reagents. Oh.
I don't know if we had reagents, but she can answer it.
Inaudible. Yeah.
Sure.
Do you need to do repetitive testing to validate your results? That's fine.
What type of testing?
Repetitive. Repetitive.
Oh.
She testified that has presumptive and confirmatory, so not sure if that's a question. Do multiple people perform various phases of the analysis?
Yeah, good question.
How do you handle chimeras? Handle what? Chimeras, chimeras.
I don't even know that is.
I know it's.
I think I've seen that word before.
Yeah. The next question is, does a generational chimera weaken over generations, or can the contributor be isolated? Are we in a rabbit hole? Are we gonna ask this question? I mean, that never came up in either of your direct, recross, redirect.
I'm assuming she would know, but I'll object to it.
I mean, I think if she can answer it, she can. I don't know.
I don't even know what the question's asking.
I don't either.
No. I just don't want to ask it.
Yeah. So just ask, how do you handle chimeras? Yeah. And then ask her.
I'm fine with that.
Are your storage refrigerators for standards locked at all times, even if inside the locked laboratory? Yeah.
That's fine.
What personnel, what restricted access policies are in place? And it says, what personnel restricted ACC, this says restricted access regarding personnel.
What personnel have access?
Can toxic drugs or inhalants alter the DNA strands? Wow. Can you read it one more time? I'm sorry. Can toxic drugs or inhalants alter the DNA strands? I think what they're getting at is if you put, like, chloroform over someone's face, can that alter the DNA? I mean, that's fine. What is PCR, an analyzer or methodology? Polymerase chain reaction. Yeah. Okay. Can we just ask what is PCR? 'Cause then she can describe. Yeah. I know what it is. I think that would be it would be. Okay.
Sidebar conference concluded.
What types of quality control is necessary in the testing for DNA reagents?
DNA reagents go through, you prepare them through a series of steps that are, a protocol that's followed, that's logged. The date and time of reagents being prepared is logged, as well. And we adhere to expiration dates from manufacturers as far as the chemical components of our reagents. So a lot of our reagents we make in-house from chemicals that are bought from manufacturing companies. And then, as far as the use of reagents, we do a quality control on their agent to make sure that it's working appropriately by testing a positive and a negative control to ensure that it's giving us the expected results when doing our testing.
Mr. Luttrell.
I have no questions on that.
Ms. Auger. No questions. Do you need to do repetitive testing to validate your results?
Within the scope of a forensic case, no, there's not a need to do like a duplicate test or reproduce a test as a confirmation of any sort. I will say that as something that is considered as part of our validation studies, we'll consider, testing multiple samples to ensure that we're getting, consistent results from a sample.
Mr. Luttrell.
And the results of those validation studies are that do you get consistent results or not?
Yeah, it's really dependent. That's kind of within the scope of a sensitivity test and a validation sample, or even like a concordance test. We'll make sure that it's yielding reproducible results in that validation. If we're reaching a sensitivity level that isn't yielding reproducible results within validation, then that's kind of how we help determine our thresholds for when we can make interpretations on a sample. So if a validation study will show you're not getting reproducible and consistent results, then that might fall outside of the scope of what is something that we would incorporate into our procedures.
Ms. Auger. Nothing. Do multiple people perform various phases of the analysis?
Not within our laboratory. So we have a case is assigned to an analyst, and that analyst works the case all the way through for the majority of cases. Occasionally, you'll have an analyst that may only do thesserological examination in a case, and so they'll do all that screening and determining what body fluids are present, and then a separate analyst may do to the DNA analysis. But for the most part, most cases in our laboratory are completed by one analyst all the way through.
Mr. Luttrell.
So if make sure I understand you correctly, are you saying that every exam that you talked about and the items that you examined in this case, that you were the only one who conducted all phases of the exams?
In all the results that we've talked about here today, I am the only that did those examinations. I didn't have an assistant or a technician or another analyst doing those testings.
Thank you.
Ms. Auger. No. How do you handle chimeras?
I think that the question is referring to chimeras, and that's a good question. So individuals that are chimeras may have a very unique DNA profile where some of their body fluids might be consistent with one profile, but perhaps. I personally have not seen that in my analysis. So if something like that were to arise, we definitely would have to, if that was something that we expected in a case, then that would be something that would have to be considered. So typically, in our past request for labs form, we would have a question for has an individual been transfused, because a transfused individual is someone who might exhibit two different DNA profiles, one from their donor and one from their own DNA. That hasn't come to mind in any case that I can think of, but in that instance, we would need to have a DNA profile from both of those kind of body fluid sources to be able to compare back to an unknown profile.
Mr. Luttrell.
Thank you. I have no questions on this.
Ms. Auger. No questions. Are your storage refrigerators for samples locked at all times, even if they are inside a locked laboratory?
Not necessarily. We have personal evidence storage boxes within a freezer. If I have evidence or extractions within that personal storage box, then it is locked, but we do consider our laboratory secure once the laboratory space is secure, so we don't always need to lock those personal evidence storage just for the refrigerator. My personal dry storage is always locked.
Mr. Luttrell.
Nothing.
Ms. Auger. No. What restricted access policies are in place with personnel?
So our laboratory is structured in a way that you have to badge to get into the laboratory, so that's monitored. Only certain individuals can get into the laboratory area itself, so only biology analysts can access that area. No one outside can get into the laboratory areas, or even individuals from other units can't access our laboratory without being escorted into our unit. As far as the overall laboratory, contributors can come into just a couple of entry points into the laboratory. If they want to get beyond those guest areas, then they are escorted at all times and don't have access to the overall laboratory, as well.
Mr. Luttrell.
I have no questions.
Ms. Auger. No questions. Can toxic drugs or inhalants alter the DNA strands?
I'm not really sure. I think that the effects of drugs could affect maybe like the morphology of cells, I've seen that where I believe like a sperm cell could be affected by that type of drug use, but I can't say whether it would affect. I don't believe, obviously, we all have cellular material in our body, but it shouldn't affect whether there's DNA that can be left behind by someone who might have had a toxic exposure to a drug.
Mr. Luttrell.
No questions.
Ms. Auger. No questions. And what is PCR?
PCR standards for polymerase chain reaction, and that's the method that we use during amplification to make millions of copies of DNA. So I know I mentioned 25 to 50 cells that need to be left behind in a sample. In order to actually visualize that little bit of cellular material, I need to amplify it, and PCR is that process in which I can make millions of copies of it.
Mr. Luttrell.
And when you make those copies, are you changing the DNA?
Essentially, no. If there's a sufficient amount of DNA present, then it's making copies of the DNA that's present. It's when you get into a low template quantity, where there's just a very little bit of DNA, then your amplification process could cause some effects that I'd hesitate to say change it, but may give you some effects that aren't necessarily accurate.
Thank you.
Ms. Auger. No. All right. Thank you, Ms. Bozinovski. You may step down.
Thank you.
You may call your next witness.
Thank you, Your Honor. The state will call Pat Cicero.
Witness sworn.
Just one moment, Your Honor, if I may select some.
Sure.
Would you state your name for the jury, please?